Hello everyone and welcome to Instant Biology by Dr. Neela. I am again here with you with another very important lecture, and the topic of today's lecture is mismatch repair system. Now you would be knowing that we are continuously making lectures on DNA repair system, and in the past we have completed topics such as direct repair system, in which we had discussed about the photo activation of the enzyme DNA photolyase. Apart from that, we have also discussed about the indirect repair system, and under indirect repair system I have talked about the nucleotide excision repair, the base excision repair, and in today's lecture I would be talking about mismatch repair system. This is an important lecture, and some questions have been asked in the previous years in CSIR, DBT, ICMR, as well as GATE, biotechnology and life science examinations. So what we would be discussing in this lecture, let me just give you a brief introduction or brief overview about that. So in this lecture I would be dealing with the process, or what about the introduction of a mismatch repair system, how is mismatch repair system performed, or what is the mechanism of mismatch repair system. I would be showing you some interesting diagrams through which you can understand the concept in a better way. Apart from that, I would also be talking about, or I would also be taking up some of the PYQs, not only of the mismatch repair system, but I would also take up some of the PYQs that have come from the DNA repair system in general, and we would do the questions. So for those of you who understand Hindi better, I have already uploaded a video on mismatch repair system in Hindi. For those of you who understand English better, so this video would be in English. In the past I have made the playlists separately so that you can understand it in a better way. So without much ado, let us start our lecture, mismatch repair system. So if firstly you can see the image that is in front of you, in the page, in the slide in front of you, you can see that there is a big DNA, and some of the workers are stationed on this platform, and what they are doing is they are going for repairing of the DNA. So this image actually sums up what we are going to discuss in the subsequent slides. So what are these workers? These workers are nothing but these are enzymes that we are going to talk about. So they are enzymes, right? So because enzymes are the major participants of a repair system, I would be discussing about the enzymes and their functions in this lecture. Now coming to the first slide, mismatch repair system. What is the purpose of mismatch repair system? Now, as I've previously explained also, that all the repair systems have an important role to play to repair the DNA, but the difference between the different repair system is that what kind of repair are they catering to, or what kind of repair are they doing, and apart from that, what is the time frame, or when is the DNA repair system, when a particular DNA repair system gets activated. So if I'm particularly talking about mismatch repair system, it detects mismatches in DNA replication process, So it checks out the mistakes in the DNA replication process. Now when I say DNA replication, what do you understand? Now let me just draw quickly over here. So let us say that this is strand number 1, this is 5' to 3'. Obviously this strand would undergo replication and a new strand would be formed like this. So this would be 5' to 3'. So this is the new strand and replicated strand. This one is the replicated strand. Now you would be knowing that when the replication takes place, there are chances that errors might get induced, or while adding new nucleotides, errors might occur. This error might be due to the malfunction of DNA polymerase enzyme, and there can be other reasons also, but what is important to understand over here is that during the process of replication there might be some errors that might happen, and if during the process of replication these errors would occur, then the mismatches, or then the errors would be identified and repaired by mismatch repair system. Now again and again I am using the word mismatch. Again and again I am using the term mismatch. Now what do you understand from mismatch? Let me just quickly make you understand. Let us say that this was the main strand. Let us say on the main strand there was an adenine present over here. So in the corresponding strand, or in the newly replicated strand, the green one is the newly replicated strand, over here thymine should be present. Obviously corresponding or opposite to adenine, because adenine would be base pairing with thymine only with 2 double bonds, so thymine should be ideally present over here. But what happens is, because of some kind of an error, let us say this thymine is not present over here and there is a mutation that occurs, and let us say this is cytosine, or let us say this is guanine. Obviously this is a mutated base, and this has occurred during the process of replication, because DNA polymerase will be synthesizing the new strand in this direction, and it might have added a faulty base. So this is cytosine that has been added, and it is a mismatch. Why am I saying mismatch? Because this is adenine, and corresponding to adenine thymine should be present, therefore it is a mismatch. So when mismatches occur during the process of DNA replication, as I have said, then this kind of mismatches are repaired by mismatch repair system. Now I have given you the introduction, I have given you the basics. When does mismatch repair system gets activated and what does it do? Now let us talk about the mechanism of mismatch repair system. So firstly, when the mismatch repair system functions, it has to first of all recognize the mismatched base pairs. It has to first recognize the mismatched base pairs. So, what do you understand from mismatched? I've already said what is the meaning of mismatched. You can see over here also, I've shown that the black strand over here, A, T, G, C, G, A, T, C, this is the original strand, and it has undergone replication. So this the lower strand, this is the daughter strand basically, and you can see that in this position, in the original strand guanine was present, and in the corresponding strand, the newly replicated strand, we can see an adenine. Obviously this adenine should not be present over here, because guanine ideally pairs with cytosine. So obviously this is a mismatch. So that is what I am trying to explain over here, is recognize the mismatched base pairs. So the first step that is performed by the mismatch repair system is to identify the mismatched base pairs. Now next one is determine which base in the mismatch is incorrect one. Determine which base. Now this again raises a question in our mind, that which base in the mismatch is incorrect one. What is meant by this? Let me come back to this particular slide. Okay, now you can see that this, in the first strand you can see over here, in the main strand you can see guanine present over here, and in the next strand you can see adenine present over here. Now the problem is, when the repair system will come at this particular point or will arrive at the scene, how will the repair system identify that which strand is the original strand, and which strand, or rather which strand is the main strand or primary strand, and which strand has been replicated recently? How will it identify? It does not have eyes. How will it identify that which is the original strand or which is the error-free strand, and which strand error has occurred? Because to someone who has just arrived to the scene, they might say that this guanine might be the erroneous base, and for some other people this adenine might be the erroneous base. You cannot identify that which is the original strand and which is the newly replicated strand. Obviously the error would be seen in the newly replicated strand rather than the original strand. The original strand would, of course, if the error was there in the original strand, it would have been catered to before only. But now, since the replication is now occurring, so the chances are that the newly synthesized strand would have the error. But the problem is, when the DNA repair system arrives over here, how will it identify that which is the original strand and which is the newly replicated strand? So this is the problem. So parent polynucleotide is the correct sequence. Obviously the parent, or the original one from which the replication is taking place, this would be the error-free one. This is the parent one, this would be the error-free one. The next one that is synthesized, or the daughter strand that is synthesized, would be the incorrect sequence, or would be the sequence in which the mutation would have occurred. Now the problem comes, how would you identify? This is clear to us that parent polynucleotide would be correct sequence and daughter polynucleotide would be the incorrect sequence. This is clear to us. Now the next thing that we have to cater to is to identify which strand is the parent strand and which strand is the daughter strand. Okay, now Important thing to identify, or how does the DNA repair system identify, is it's very simple. The concept that it uses is very simple. Actually what happens is, in the original strand or in the parent strand, the methylation is seen on certain bases. Now you can see over here there is an adenine present over here. Now this adenine is methylated in the parent strand, but the adenines are not methylated in the daughter strand. So you can see that this daughter strand does not have methylated adenine. So you can say that it is under methylated. That means it is not having the methyl groups on the adenine, but the parent strand is having the methyl groups on the adenine. So this actually provides an identifying feature between the parent strand and the daughter strand. So DNA repair system looks for this difference. Now one thing can come to your mind, that are all the adenines methylated in case of the parent strand, and are all the adenines non-methylated in the daughter strand? I would give the answer to you in just a moment. Coming back to our original slide, so determine which base in the mismatch is the incorrect one. So again you got the answer to this by simply looking at which is the methylated strand. Methylated strand would be the parent strand and non-methylated strand would be the daughter strand. Now the next step is excise incorrect base and carry out repair synthesis. This means that the incorrect base or the faulty base is taken out from here, nitrogenous base is taken out from here, and the correct base is brought in, and the repair synthesis is performed. So this is the third step. So this was the first step, second step, and this is finally your third step. Okay, now we have understood this slide. In this I have just told you that which or how will you identify the parent and how will you identify the daughter strand. Now understanding that which adenines will be methylated, this is important. Always remember that it's not like that all the adenines on the parent strand would be methylated. No, actually there is a particular sequence, and this sequence is identified by particular enzymes. So there is a particular sequence, and this sequence is identified by particular enzymes. For instance, there is a sequence, as you can see in your screen, that there is a sequence which says that GATC. Now this is a sequence GATC. Now whenever there is a sequence GATC on the parent strand, this parent strand, or I mean to say whenever there is a sequence GATC, a enzyme called as DNA adenine methylase, you can see the name of this enzyme, DNA adenine methylase. So in short it is also called as DAM. So what will happen, this adenine that is present over here, this adenine will be methylated. On which position? It will be methylated at the 6th position. That means if you remember the structure of adenine, then on the 6th position this adenine will be methylated. So that means whenever all the adenines are not methylated, actually only the adenines that are present in this sequence GATC, wherever GATC are present together, that adenine will be methylated at the 6th position. Similarly, cytosine also gets methylated. Now it will get methylated by a different enzyme. The name of the enzyme is DNA cytosine methylase. Name of the enzyme is DNA cytosine methylase, DCM. Okay. Now where will it add the methyl group? Obviously on cytosine. But what is the sequence? I am talking about the sequence. So CCAGG, this is the sequence, and CCTGG. So wherever these sequences are present, CCAGG, CCTGG, wherever these two sequences are present, obviously the cytosines present over here will be methylated. So cytosine is methylated to 5-methyl cytosine. So these cytosines are present over here, and these cytosines are methylated to 5-methyl cytosine. So now you have got a clear understanding that which adenines or which cytosines will be getting methylated, along with which enzymes are responsible for methylating these particular bases. So this would be clear in your mind. Now let us move forward and understand this. Many of you might understand, or many of you might think that methylations might prove to be mutagenic in nature, because actually what we are doing is we are altering the structure of the bases. You are creating methylation. But no, you are wrong if you think so. Why? Because methylations, whatever methylations we are doing or whatever methylations are done in order to identify or differentiate between the two strands or the parent strand and the daughter strand, they are not at all mutagenic, so they would not create any problem with the mutations. So there will be no problem with respect to mutations. Next, let us come to another problem or another confusion that many students have, is that when there is a parent strand and a daughter strand is created out of this parent strand. So this is the parent strand, and from the parent strand a daughter strand is created. Now, is it like this? The daughter strand will never get methylated and only parent strand will get methylated? Is it like that? No. The answer is no. Just after the replication completes, there is a very small window of time in which the DNA repair system has to identify between the daughter and the parent strand. Why is that so? Because after this time period or after this small window gets over, the daughter strand will also get methylated in the same way as the parent strand was methylated. That means DAM and DCM enzymes will come and start methylating the daughter strand also. That is why I am again and again saying that there is an extremely small window between the methylation, in which the repair system or mismatch repair system have to identify the mismatch, have to identify the incorrect sequence, and have to start working on it and correct it. Because after that particular time period, the parent strand and the daughter strand would be resembling each other with respect to the methylation. Both would undergo methylation. Okay. Sorry. Now, so there is a delay between DNA replication and methylation of daughter strand. So during this time, the mismatch repair system works. This is what I am saying. Okay. So only during that time window, the mismatch repair system has to work. Okay. Now, 3 mismatch repair systems in E. coli have been identified: long patch repair system, short patch repair system, and very short patch repair system. So only 3 different types of repair systems have been identified. What we will be discussing majorly, the long patch system is more often asked, and it is the important part of our syllabus. So we would actually be discussing only this portion, in which I would be talking about long patch system, or how does mismatch repair system function in long patch repair. So let us talk about the enzymes that are involved over here. Only 3 MUT proteins are involved. That means 3 enzymes or 3 proteins are involved over here that would be taking care of the entire repair system. Now this is extremely important. You should be knowing about the 3 proteins and their functions. Let us first of all talk about the first protein, and the name of the protein is MutH protein. What is the purpose of MutH? There are 2 main purposes of MutH. The first purpose is to recognize GATC. Why do you want to recognize GATC? Because again, the adenine over here will be methylated. So this MutH will be identifying GATC. Wherever the GATC is present on the 2 strand, it will be identifying those strands. It will be identifying the GATC region on both the strands. And then it will check out which strand or which GATC is methylated and which GATC is not methylated. Of course, the GATC that is not methylated is our newly synthesized daughter strand, and now this only has to be corrected. So recognition of GATC, and then it also functions as endonuclease enzyme. What do you mean by endonuclease? Endonuclease means the enzyme that can create cuts in between the sequence. In the middle of the sequence, it can create cut. Now coming to the next enzyme and next protein, and this protein is called as MutS protein. Now what is the purpose of MutS? Recognition of mismatches from the distortion they cause in the DNA backbone. What I mean is, obviously GATC would be recognized by MutH. Now MutS, what function does MutS have? MutS will be recognizing the mismatches from the distortion, because there will be a mismatch obviously GATC will be telling us that which is the original strand and which is the parent strand and which is the daughter strand. But where is actually the mismatch present, where is actual problem present in the strand, that will be identified by MutS. How come? Because whenever there is a mismatch, suppose adenine obviously pairs with thymine, but suppose adenine has on the next strand or on the complementary strand, adenine is now pairing with cytosine. Obviously the proper pairing will not take place. That is, let me just draw over here. So let us say that this is my original strand. Let me just check out if you are able to see this or not. Yes. So this is my original strand, and you can see that this is over here adenine. When I synthesize my new strand, it's like this. Obviously thymine should be present over here. If thymine is present, then there will be a double hydrogen bond. Two hydrogen bonds would be pairing adenine and thymine. But let us say that thymine is not present over here and there is a mutation, and because of some reason cytosine has been attached over here. Now you obviously know that adenine and cytosine cannot pair with each other. So what will happen? There will be a distortion. There will be a distortion, something like this. There will be a distortion. So this distortion only is recognized, this distortion in the DNA backbone is recognized by MutS enzyme. Also, this MutS has ATPase activity. That means it can utilize ATP and it can perform a particular function. So distortion is identified by MutS, GATC is identified by MutH, and let us talk about MutL. MutL has unknown function. We still do not know about the functions of this. So this is very, very important slide. You should of course remember this. Now let us move on to the next point. MutS, since I have discussed about MutH, MutS and MutL over here, let me give you some more important points related to MutH, S and L. Now MutS actually recruits MutH and MutL. This means that MutS is the first enzyme that actually reaches to the point. So after MutS reaches to the point, because MutS will be identifying the DNA distortions, and when it will observe DNA, when it will be observing distortions, only then we have to check that which strand actually is the parent and which strand is the daughter strand. So firstly MutS reaches that particular spot and it identifies that where actually is the lesion, where actually is the mismatch. After that it will recruit the MutH and MutL. MutH, as I have already said, that it will function as an endonuclease. It will cleave unmethylated strand. Obviously, what do you mean by unmethylated? By unmethylated, I simply mean daughter strand. So it will be cleaving unmethylated strand on the 5' end of G in GATC sequence. Now this is again an important thing. So the lesion, wherever the mismatching is present, close to this mismatching, the GATC will be identified, and the MutH, which functions as an endonuclease, it will cleave unmethylated strand on the 5' side of G. So suppose this is a lesion over here, this is a lesion, and let us say that this is your GATC, this is your 5' end, this is your 3' end. So it cleaves unmethylated strand on the 5' end of G. 5' end of G. 5' end means G over here. So it will be cleaving over here. So the strand will be cleaved over here and it will be cleaved from here till here. Of course, a greater region will be removed that will also be having the lesion. This is the lesion. This is the problematic area. This is the lesion. Okay, so cleaves unmethylated strand on the 5' side of G in GATC sequence. Next, DNA helicase 2 plus exonuclease 1. These are important enzymes. DNA helicase 2 and exonuclease 1 removes the segment between cleavage site and a point beyond the mismatch. Now they have mentioned two more enzymes over here. The name of the enzyme is DNA helicase and exonuclease 1. MutH just created a cut. So this cut over here was created by MutH. But MutH cannot remove the entire strand. It cannot remove the entire this black region. It cannot remove the entire faulty region. So there are further two more enzymes that are called up, and the name of the enzyme is DNA helicase over here and exonuclease 1. Both of these enzymes remove the segment between cleavage site and a point beyond the mismatch. So let us say that this is a point beyond the mismatch. So this entire region is cleaved out from here by the two enzymes DNA helicase 2 and exonuclease 1. Remember this. So what does the helicase enzyme do? Helicase enzyme will break the hydrogen bonds between this strand and the next strand that would be there. And exonuclease, what it will do? It will cleave again, just like endonuclease, but just exonuclease 1, it will be cleaving at the end of the strand. So one cleavage is over here, one cleavage, let us say, is somewhere over here. So all the entire strand would be removed. Next, when the entire strand is removed, then gap filling also has to be performed. We have to perform the gap filling. The gap filling is performed by DNA polymerase and sealing is done by ligase. So important point is, when the faulty region is removed, so this region, this gap is filled up by DNA polymerase enzyme, and then again the portion is sealed up by DNA ligase. We have discussed this multiple times. Now let me just show you with the help of a diagrammatic representation. This will make the entire thing clear to you. Now you can see that this is a 3' end, 3' to 5', and this is 5' to 3'. Okay. Now you can see that this is a methylated DNA strand. This is methylated. You can see a methyl group present over here. CH3 group is present. Wherever CH3 group is present, we can say that it is methylated, and where CH3 group is not present, it is unmethylated. Unmethylated is daughter strand and methylated is the parent strand. Next, which enzyme will come first? MutS will come first. What is the purpose of MutS? MutS will recognize mismatches. Once the MutS recognizes the mismatch, and mismatch is present over here, so mismatch will be recognized by MutS. The green thing, this green circular thing is your enzyme. It is MutS. It is recognizing the mismatch. Next, MutH will be recruited now. MutH, what it will do? It will bind to the methylated A. It will first of all identify that which strand is methylated, which strand is not methylated. It will bind to the methylated A region. So you can see that this is GATC, right? So on the GATC, it will bind to the methylated A. That means this methylated A, it will bind to the methylated A and links with MutS through MutL. So this is MutH, this is MutS, and MutL will be causing a bridge or link between the two. Now MutH nicks and exonuclease excises a short segment of a newly synthesized strand. What I want to say over here is MutH, because it has a function that it will MutH nicks and exonuclease excises. MutH will be nicking and exonuclease excises the short segment of newly synthesized strand. The newly synthesized strand is the short segment of the newly synthesized strand is removed, and then this is the gap that is created. The gap that is created is filled by DNA polymerase, and the excised DNA region and DNA ligase will be the excised DNA region is removed. The gap is filled by DNA polymerase, and ligase will be coming and just making the phosphodiester bond, so that strand is completed. Now this is the case. Okay. Remember, if you see, this MutH had the function of endonuclease. So that is what I have written over here. So MutH will be creating a nick or endonuclease. Basically exonuclease and DNA helicase will be cleaving of the strand. It will be removing the strand. So this is what we had discussed up till now. So this is what happens. Majorly you should be knowing about the functionality of MutH, MutS and MutL. Apart from that, DNA polymerase and DNA helicase 2 also are important. Apart from that, you should also know that this entire process occurs in case of replication, whenever the replication is occurring. So this is the entire thing about the mismatch repair system. Now let us talk about some of the PYQs. Now these PYQs would be related to DNA repair system, of course. and the first one is the mismatch repair activity of E. coli repairs misincorporated bases which is not removed by proofreading activity of DNA polymerase. However, while doing so, it has to decide which strand of the DNA is newly synthesized. This is what we have been talking about. Which strand of the DNA is newly synthesized and which one is parental. That means which one is daughter and which one is parental. Mismatch repair system does it by which of the following ways? Basically, it will be doing it by, it recognizes nearby GATC sequence. Yes, it does recognize, but obviously it recognizes the methylated sequences. It recognizes nearby palindromic sequences. No, this is not true. It recognizes specific repetitive sequence. No. It recognizes hemimethylated GATC sequence. Yes, correct. This will be the correct answer. So correct answer is 4. Okay. Now let us move on to the next point. If DNA, if thymidine is replaced by uracil, then what would be drastic changes in the organism? Now, if you understand that thymidine, or rather we should say that cytosine, when it is demethylated, it is just like uracil. That means the appearance of both of these nitrogenous bases would be same. That means cytosine, when you are demethylating, it will be creating uracil. Okay. And it is seeing that, and you already know that cytosine is present in DNA and in uracil is present in RNA. Furthermore, the complementary base to uracil in DNA is thymine. So what is the problem? If uracil is attached in the DNA, then what is the problem? The biggest problem would be repair mechanism. Why? Because repair mechanism for deaminated cytosine will not work. Because the repair system will not be able to differentiate if the cytosine has been deaminated. So the cytosine, if it is deaminated, then also uracil is formed. But if you are putting in uracil in the DNA itself, how will it recognize that this uracil that is in the picture, whether it has occurred because of some kind of error that involves deamination of the cytosine, and then this uracil has been created, or this uracil is the original uracil? How would you identify that? So it would be very difficult to identify for the DNA repair system. That is why the correct answer over here is repair mechanism for deaminated cytosine will not work, because it will always understand that this uracil is the original uracil and this has not occurred due to the deamination of cytosine. Next question is, which enzyme is involved in direct repair mechanism against ultraviolet induced DNA damage? So we have talked about this in my previous class. Unfortunately, we did not do the question at that time. So ultraviolet induced DNA damages are actually treated by DNA photolyase enzyme. It utilizes blue light and it is involved in direct repair system. Direct repair system basically is called direct repair system because no phosphodiester bonds are broken. No phosphodiester bonds are broken. Next thing is UVB induced damage of DNA by formation of pyrimidine dimers are repaired by photolyase on absorbing. As I've already said, DNA photolyase enzyme works by utilizing blue light. So the correct option is blue light. Okay. Next. Pyrimidine dimers formed due exposure to UV rays can be directly repaired without removing any nucleotide by repair mechanisms known as. Now, obviously, this answer is photoactivation. Why? Because in my previous lectures also I have told you that photoactivation is a process that involves direct repair system, and in one of the previous questions I told you that direct repair system is the repair system where there is no breakage of phosphodiester bond. So we are not removing any nucleotide, we are not breaking any bonds. So they are saying that without removing any nucleotide by repair mechanism, we are not removing any nucleotide over here. It's just that repair exists without removing or without breaking any bond, phosphodiester bond. So this is called as photoactivation process. Coming to the last, so these were the questions that I wanted to take up. I hope that you would have enjoyed the session. Just one quick announcement is that currently we are running our GAT-B batches. GAT-B batches are in full swing. So the charges for this batch is just rupees 3500, and in these charges you would be getting the complete notes, the recordings of the lecture. Apart from that, you would also be getting the DPPs, you would also be getting the test series, and we are also dealing with the part A questions, so that would also be an added benefit to you. So if you are interested, you can always connect with us at 8299134528. You can connect with us on WhatsApp. Apart from that, if you want to connect with us through email, the email has also been mentioned in the description section. Okay, so thank you so much and have a good day.